Bismillahirrahmanirrahim. I am Tariq Shahzad and now we are going to discuss about the reproduction in fungi, that how fungi reproduce and it increase in number and disperse itself throughout different environment. And in this we will discuss only asexual reproduction, that how fungi reproduce asexually and it increase in number. Through asexual reproduction, fungi have 4 different methods. Fungi can reproduce asexually by spores formation, by conidia formation, by fragmentation and budding. These are 4 different methods of asexual reproduction. We will discuss it in detail. This is a long question. So in first paragraph he says that fungi reproduce sexually mostly and asexually less. That one is also an MCQ. That first thing, keep in mind that there are both methods of reproduction that are found in fungi. Fungi can reproduce through sexual reproduction as well as it can reproduce through asexual reproduction. Both methods are found, but sexual reproduction is mostly. This one is an MCQs, and asexual reproduction is less. So mark in your book that sexual fungi reproduce mostly through sexual reproduction. Means there are both type of method in fungi, sexual reproduction and asexual reproduction, but sexual reproduction is mostly observed in fungi. Next one is no sexual reproduction is seen in imperfect fungi. No sexual reproduction is seen in imperfect fungi. No sexual reproduction mean that's why we say that it is imperfect fungi. Perfect and imperfect. Perfect mean complete and imperfect mean incomplete. In all other type of fungi there is both sexual and asexual reproduction, but in case of this fungi, imperfect fungi, its other name we will also discuss detail in the next topic, that it is incomplete or imperfect. Why? The reason is that there is no methods of sexual reproduction in imperfect fungi. This one is an also MCQ written in this paragraph that we are discussing now. Next one is asexual reproduction. Our question starts from here, that what are the different methods of asexual reproduction in fungi. This one is an long question. So to discuss asexual reproduction in fungi, first of all you have to write the all 4 names of method through which fungi can divide can reproduce asexually. So these 4 methods you can write their names according to your sequence. You can change their position, and one method is spores, other method is conidia, and third method is fragmentation, fourth method is budding. These are 4 different methods through which fungi can reproduce asexually. So we will discuss first of all the spores. In the last line of your topic written is that common means of reproduction. Means spore formation is a common means of reproduction. This one is an MCQs. And next one is spores produce inside sporangia, and if there is only single sporangia, it is known as sporangium. Sporangium. Spores are most common means of reproduction and they are produced inside sporangia. Means if it is single, then it is known as sporangium. Next, this is the diagram of sporangia. That is single one, sporangium. Here there are different spores that are present within the sporangia, and the red dots within the sporangium shows that these are spores, and they are attached with the rhizoids, and that one is hyphae. And you see that the sporangia and its bearing hyphae is completely cut off from the other hyphae or main hyphae through a septa. And my daughter is crying. Wait for some time. Yeah, next one is there are different properties of spores. That what are the properties of spores? Spores may produce sexually, spores may produce asexually. These spores are haploid and non-motile, small in size, large in number, not need water for their dispersal, dispersed by wind, insects, small animals, and rain splashes. And whenever they dispersed and land on a suitable place, they will germinate to give new mycelium. They will germinate to give new mycelium, and that is the life cycle when fungi reproduce, forming spores through asexual reproduction. That one is a mycelium, and on which a sporangium is attached, and sporangium bears spores. These spores disperse, and when land on suitable soil, they will germinate and mycelium is formed, and again this mycelium will change into this form and produce sporangia wherein spores are formed, and this cycle will be repeated. So that was all about the reproduction in fungi through spores formation. Once again, through spore formation, spores it is a common methods of asexual reproduction in fungi, and these spores are produced within the sporangia, not outside the sporangia. They are produced within the sporangia, and these spores may be produced by sexual reproduction, asexual reproduction, large in number, small in size, and whenever they dispersed, they only dispersed by wind, rain splashes, small animals, insects, but not they needed water for the dispersal. And whenever they land on a suitable soil or on a suitable medium, then they grow to form their mycelium again, and their sporangia are formed in which spores are produced, and then the cycle will be starts. So you can see in this diagram that all things are haploid, no one is diploid. Diploid is only related to sexual reproduction. In asexual reproduction, fungus cells are also haploid, so there is no chance of their fertilization, because the reproduction is asexual. So in asexual reproduction, there is no chance of their fertilization, so no haploid structure is seen in this diagram. And next one is fragmentation. Fragmentations mean part formation. They break into different parts, and these parts can form the mycelium. Means whenever mycelium is mature, and whenever this mycelium is mature, it will be broken down into different pieces or into different parts, and these parts will germinate, or these parts will grow to form a new mycelium. So in fragmentation, it says that mycelium break, and each fragment give rise to new mycelium. For example, that one is a total mycelium, and it will break into different parts, azanic being called. So we will start after this. Yeah. Next one was budding. That how they reproduce by budding. Budding mean bud formation, and bud is an outgrowth. Outgrowth mean a part that show more growth, and the nucleus of the parent will divide, and some of the nucleus goes to the bud. Bud increase in size, and when it attain the size a size equal to the parent cell, then it have both option. Either it detach from the parent or can attach with parent to form a filaments or large colonies. So in budding, bud outgrowth on the yeast. That process is seen in yeast, and there are two unique features about yeast. Yeast is a non-hyphal, and yeast is a unicellular fungus. So asymmetric division, that one is an MCQs, are equal division. It means that in both type of division, asymmetric mean unequal and equal divisions may occur. That one is an MCQ, that asymmetric division is occurred in budding, not in any other type of reproduction. Next one is bud detach or attach to parent. Mean both options are available. Either it detach from the parent, means separated from the parent, or it can combine with them, or live attached with their parent atom and form a large colonies. So that is the yeast cell, and a bud is formed, nucleus divide and goes into bud increase inside, and other bud is formed. It means that at a time more than one bud can be formed, and next the bud is separated, first bud and second bud also going to attain the size equal to their parent cell. So that was all about the budding. And last one is conidia. Conidia are asexual spores. It is also an MCQ, that conidia is an asexual spores. So asexual spores are conidia, non-motile, produce on conidiophore. Produce on conidiophore mean not inside. That one is also an MCQ, that conidia produce outside the structures, not inside. Produce on conidiophores, not inside, in the form of chains, cluster, large in number, survive weeks. It means that it can survive many weeks. So conidia have more resistance to cope with the harsh environment or unsuitable environment. So means besides other methods, the methods of conidia formation make the help for fungus to survive in unsuitable or undesirable condition. So large in number and survive in weeks. That one is in the diagram of conidia. These are spores that are called conidia, and they are produced on the conidiophore. That is conidiophore on which the conidia are present. So that was all about the asexual reproduction in fungi. Sexual reproduction in fungi. Our last topic, in our previous topic we were discussing about asexual reproduction in fungi, and you know that there are 4 different methods of asexual reproduction in fungi, but today we will discuss about sexual reproduction in fungi, that how fungi reproduce sexually. So sexual reproduction in fungi is simple than asexual reproduction. In sexual reproduction, as we know that there are 4 different group of fungi: zygomycota, basidiomycota, ascomycota, and imperfect fungi. But in imperfect fungi, the process of sexual reproduction is absent. It means that then only there remain 3 different group: zygomycota, ascomycota, and basidiomycota. In these 3 groups, the sexual reproduction has been observed. So in these 3 group, the sexual reproduction proceeds in different way. There is a slightly variation in the reproduction methods in them, but the thing that is common in all these 3 group is that there is a fusion of nuclei in all different group, followed by the process of meiosis in which spores are formed. So the process of sexual reproduction is somewhat different in all 3 group, but besides this difference, the common thing in all 3 groups is that there is a fusion of nuclei, number 1, and number 2, the formation of spores after the formation of spore by the process of meiosis. So that is the common thing in all these 3 things. So we start from here that In sexual reproduction in fungi, that one is a long question. Fusion of nuclei occurs means two different nuclei come close together and they intermix to form the diploid zygote. And number two, different in all three groups. But the common thing is that fusion of haploid nuclei followed by meiosis. Fusion of haploid nuclei and meiosis takes place in all groups. In all groups, this process takes place. Next one is process of sexual reproduction, that how this process is performed. The process is so simple that there are two different hyphae that is only genetically different, but compatibility means they are genetically different but they are compatible, they can intermix with each other. When they come close to each other, then first their cytoplasm fuse together and then their nuclei fuse together. First cytoplasm fuse together, this is known as plasmogamy. After this the nuclei come close together, that is known as karyogamy. So between karyogamy and plasmogamy, or plasmogamy and karyogamy, there is two different groups in some fungi. For example, zygomycota, the fusion of nuclei after the plasmogamy takes place immediately. But in two groups, ascomycota and basidiomycota, the fusion of nuclei after the plasmogamy does not take place. It takes too much time after the plasmogamy. So that is difference between them. The process is that hyphae of two genetically different but compatible, mean they are genetically different. There are two different nuclei. As we are saying that there are two different hyphae, it means that the hyphae are formed from the spores and spores are produced through the process of meiosis in sexual reproduction. So these hyphae are different. So these are genetically different but compatible, come close together. First their cytoplasm fuse, then nuclei. First their cytoplasm fuse, then their nuclei fuse. And if their cytoplasm fuse, it is known as it is the first step and that is called as plasmogamy, whereas in second step, that is karyogamy, the fusion of nuclei occurs. That one is a short question that differentiate between plasmogamy and karyogamy. Plasmogamy, the fusion of the cytoplasms, is known as plasmogamy, whereas the fusion of the nuclei is known as karyogamy. So in karyogamy, the fusion of nuclei, there are two different groups, basidiomycota and ascomycota. Karyogamy is late, mean in basidiomycota and ascomycota, the fusion of nuclei does not take place immediately after plasmogamy. In karyogamy, after the plasmogamy, there is a difference, or there is a time required for this process to complete. So in these two groups, basidiomycota and ascomycota, the karyogamy is late. That one is an MCQ. Acha, it means that if the karyogamy is not taking place immediately after plasmogamy, it means that now the hyphae are single, and in this hyphae two different nuclei are formed and two different nuclei are present. These nuclei not only present, coexist, they can divide within the same hyphae, and this hyphae bearing two genetically different nuclei will called as dikaryotic hyphae or heterokaryotic hyphae. But all this process we can summarize in a simple diagram, that this one is one type of hyphae and this is another type of hyphae. In this type of hyphae we represent it with an other ink, and this one is represented by a different ink, that you can understand that these two are genetically different hyphae. So these hyphae come close together, it means that their cytoplasm combine with each other, their cytoplasm fuse together, and when their cytoplasm fuse together, it means that it will form plasmogamy, that is plasmogamy. After plasmogamy, their nuclei also fuse. When their nuclei fuse together, then this process will known as karyogamy, or we called it as a karyogamy. This will known as karyogamy. Now the zygote is formed. Zygote is diploid. Why? This hyphae was haploid. This hyphae was also carrying the haploid nuclei. When these nuclei will combine together, they form a diploid zygote. That zygote is diploid, and when this diploid zygote germinate and meiotically it will produce spores, and spores are haploid. As you know that during the process of meiosis the number of chromosomes reduced to half. So number of chromosomes reduced to half through the process of meiosis. That's why from the diploid zygote haploid spores are formed. Now keep in mind that these spores are formed through the process of meiosis, and we know that in the process of meiosis crossing over takes place, and whenever crossing over takes place it means that there is a variation in their genetic makeup. So these spores not resembling to this one and also to this one, but these are totally different from all their parents. Why? The reason is that they are formed through the process of meiosis. These spores whenever land on a suitable place or on a suitable position where moisture is available and food material is also abundant, then they can develop or grow to form a new hyphae or new mycelium. So that was all about the theory that how the hyphae or fungi reproduce through sexual reproduction. And in last, where karyogamy is not immediately, mean on those type of fungi, as we know that there are two different group, ascomycota and basidiomycota, where karyogamy does not occur immediately just before the plasmogamy. So the hyphae is the same, but it contains two genetically different nuclei. So that hyphae will known as dikaryotic hyphae or heterokaryotic hyphae. Where karyogamy is not immediately after plasmogamy, two genetically different nuclei coexist and divide within the same hyphae. This hyphae is called dikaryotic. This hyphae will called as dikaryotic hyphae or heterokaryotic hyphae. We will call it as heterokaryotic hyphae or dikaryotic hyphae. This one is also an short question, but here is an MCQ that a dikaryotic hyphae, he says that dikaryotic hyphae of two nuclei of different genetic makeup, similar genetic makeup, or any other options. So these two nuclei that resides within the same hyphae, if it is after the plasmogamy, then these nuclei are totally genetically different from each other. Next one is meiosis in zygote of basidiomycota will produce basidiospores. Basidiomycota, basidiospore, ascomycota, ascospore. But meiosis in zygote produce basidiospores, and meiosis in ascomycota also produce ascospore. Sometime produce, means in some cases not at all. In some cases the spores of Basidiomycota, basidiospore, and ascomycota, ascospore are produced basidiocarp and ascocarp respectively. Mean carp mean fruiting bodies are the reproductive structures, fruiting bodies that basidiocarp in some time basidia or basidiospores are produced within basidiocarp, and ascospores are produced in ascocarp. That was a long question, and I try my best to make you understand that what is the correct way to write this question. And this is a very impressive and very explanatory, and it is easy for your examiner to check your question. So try your best to write in such a way that and inshallah you will get maximum marks in your exams. Allah Hafiz.